Hello and welcome to my academy. Today we are going to talk about the immunoglobulins or antibodies. Immunoglobulins are the protein component that circulate in the blood and recognizes the foreign substances like bacteria and viruses and then eliminates them. The antibodies are produced by the special types of cells called the B cells. Today we are going to discuss in a brief about the immunoglobulins or antibodies, about their structure, about their function, and many more. Okay, without wasting any time, let's get started. Immunoglobulins, structure and function. Antibodies, the antigen binding glycoproteins, are synthesized exclusively by B cells and in billions of forms, each with a different amino acid sequence and a different antigen binding site, collectively called immunoglobulins. They are among the most abundant protein components in the blood, constituting about 20% of total protein component in the blood plasma. Antibodies are present on the B cell membrane and also are secreted by plasma cells. Basic structure of antibody molecule. The simplest antibodies are Y-shaped molecules with two identical antigen binding sites, one at the tip of each arm of the Y. Antibody has a common structure of four polypeptide chains, two identical light chains and two identical heavy chains. You must have come across the terms Fab and Fc in the immunology. To understand these terms, we need to look at a key experiment which reveals the antibody structure. Proteolytic treatment of immunoglobulin with enzyme papain and pepsin. When antibody is treated with papain, papain is a proteolytic enzyme. As the name, it cleaves the proteins. We know that immunoglobulins or antibodies are also proteins. When antibody is treated with enzyme papain, the antibody splits into three fragments of about equal size. It was found that enzyme papain cuts the antibody molecule just before the two disulfide bonds are at the hinge region, which links the two heavy chains together. As a result, two identical fragments are produced. Each of these consists of one light chain and one heavy chain held together by interchain disulfide bonds. Since these fragments have antigen binding properties, thus these are called Fab regions or fragment of antigen binding. So there are two Fab fragments contain V regions and thus binds antigen. The third fragment has no antigen binding activity. It consists of constant regions of heavy chain held together by disulfide bonds. It was found that this fragment crystallized during the cold storage. That is the reason why it's called Fc region or fragment of crystallizable. When antibody is treated with beta mercaptoethanol, it was found that antibodies contain disulfide bond. Beta mercaptoethanol is a reducing agent and breaks the disulfide bonds. As a result, all disulfide bonds are broken, leading to the split of immunoglobulins into two identical chains, that is, heavy chain and light chain. This was all about the structure of antibody. If you like the video, then do subscribe and share. Thank you.